Hello, this is Cells Part 2. Okay, central dogma of biology. We've reached a point in science where it's possible to find out all the letters in your own DNA sequence, and that's pretty cool. But if you've ever looked at a DNA sequence, you might have noticed it isn't exactly written in English. So we can find out all the A's, the G's, the T's, and the C's in a human genome, but it's a completely different language than the one that you likely learned. So how can you tell which of these letters made up the color of your eyes or the size of your ears? So fortunately, your cells can read your genome language just fine, though it does take a few steps. Your DNA tells your cells what proteins to make. So that's what DNA is. DNA is a recipe or instructions to build the proteins that make up your body. Remember that it does go through the central dogma where your DNA is first transcribed into RNA. DNA is a huge manual of instructions, so transcription is like writing down one page of DNA instructions at a time. There are 3 types of RNA made during transcription: messenger RNA, transfer RNA, and ribosomal RNA, and we'll learn more about that in our transcription and translation chapter. This is really just an introduction so that you can understand the importance of a ribosome. Okay. You'll remember that proteins are built from several different amino acids, and those amino acids, everything depends on what order those amino acids are in. So after that refresher, it's time to introduce you to the smarty pants of the cell who is able to speak that DNA language, and that is the ribosome. The ribosome is a cellular structure that performs translation or protein synthesis. Ribosomes are both language translators and efficient assembly workers. Putting together any sort of protein is like putting together a piece of furniture that has 3 boxes and more than 100 parts. The instructions are in a foreign language and has no pictures. We've all tried to do it and it's not awesome. But for ribosomes, this is what they do. Okay, they read an mRNA sequence. They translate it using tools to build it into a protein. This protein is then used by the cell, every cell in our body, most cells. Specifically, ribosomes use tRNA as the tools, and this helps bring the correct amino acids into the ribosome to build the protein. Ribosomes are composed of RNA and proteins. They exist free in the cytoplasm until they receive an mRNA message to translate. When they receive it, they waste no time in starting to read that piece of RNA and build a protein. So ribosomes can either be free in the cytoplasm or they are bound. Okay, so in the case, some ribosomes will attach themselves to another cellular structure, and that's the endoplasmic reticulum, and that is when they are bound. Okay, here these are called bound ribosomes, and they proteins produced by bound ribosomes are usually destined for a kind of specific structure, or they're meant to exit the cell through exocytosis. So in the last lecture, Cell Part 1, we looked at the endoplasmic reticulum, which is where the bound proteins are, right? They're bound to the walls of the rough ER, which is why it's called rough. And so proteins that are made in free ribosomes in the cytoplasm are going to be proteins for the cell that are in general for the cell, and then the ones that are made in the rough ER are specifically made to either exit the cell through exocytosis or made for a specific structure. So ribosomes can be distinguished into 70S ribosomes or 80S ribosomes. The 80S ribosomes are found in the cytoplasm, whereas 70S types are found in the mitochondria and in chloroplasts. So the 70S type are smaller, 80S are bigger. However, prokaryotes contain only one type of ribosome, 70S. So this is something that Cambridge really wants you to know, and it's really important that you understand. 70S are smaller. They're in prokaryotes, mitochondria, and chloroplasts, and 80S are larger, found in other eukaryotic cells.
Math and science and ribosomes. What are ribosomes? Ribosomes are small protein factories found in cells. They are located in the cytoplasm and on the ER. See the small dots on the ER and in the cytoplasm? These are ribosomes. Here is how a ribosome makes a protein. In the cytoplasm, DNA is transcribed to RNA. The ribosome reads this RNA in triplets, and the correct amino acids are put together to create a protein. Ribosomes are found in plant, animal, and bacterial cells. Why is the protein important that the ribosomes make? Let's long strands of amino acids. There are 20 types of amino acids in your body to create proteins. What are some of the jobs of proteins? Some proteins, like collagen, are used as building blocks and structural support for hair, nails, skin, ligaments, and tendons. Some proteins are enzymes, which help speed up chemical reactions. Some proteins help with digestion. Pepsin is used in your stomach to help with digestion. Some proteins move molecules from one place to another. Hemoglobin, found in blood, helps transport oxygen throughout the body. Proteins also help in muscle contraction. The protein actin and myosin are both involved in muscle contraction and movement. Some regulate body function. The protein insulin regulates glucose metabolism by controlling the blood sugar concentration. Antibiotics are proteins produced by the immune system to help remove foreign substances and fight infections. As you can see, the proteins produced at the ribosomes from the DNA converted to RNA help the cell and body stay alive and function properly. Thanks for watching Moo Math and Science Uploads a new math, science, or motivation video every day. Thanks for watching.
Okay. So it's important to remember that ribosomes, as you can see in this picture, ribosomes are built of two units, okay? A large subunit and a small subunit. Forgot to mention that. Okay, so moving on to mitochondria. Mitochondria are composed of two membranes. They're extremely important organelles within cells. Their main function is aerobic respiration, which produces ATP. So let's talk about them specifically. So they're composed of two membranes. Both are phospholipid bilayers, like the membrane around the cell, right? Like the cell membrane. The outer membrane is selectively permeable, which controls which things are coming in and out. This membrane comes complete with integral membrane proteins and pores for transporting molecules inside and out, just like the cell membrane. Now, the inner membrane of the mitochondria folds inward to form these extra surfaces for cellular respiration. And just like we've talked about in other lessons, this is what is creating more surface area so that more can be done in a smaller space. In this way, the inner membrane creates a lot of space in a small area. So unlike the outer membrane, the inner membrane does not have pores or channels and is very impermeable. Okay? The cristae are the folds created by the inner membrane. As you can see in this diagram, the surface area of the cristae can be several times larger than the perimeter of the mitochondria itself. Cells that need more energy can have more mitochondria within their cell. The cristae contain proteins and molecules that are used for making chemical energy for the cell. And then lastly, there's the matrix, which you see here. The matrix is the inside of the mitochondria, created by the inner membrane. The matrix contains enzymes for cellular respiration, as well as its own ribosomes and DNA needed to create some of the proteins important for this process. Okay, so the first stage of the process is called the Krebs cycle, and this takes place within the matrix. The last and final stage is the oxidative phosphorylation, which is a mouthful, and takes place on the membrane of the cristae. Okay, so really it's about chemical energy conversion, okay? So the mitochondria is taking the broken down monomers and small units of the food that you're eating and turning it into chemical energy for all of the cells in our body. Okay, so mitochondria are in nearly all eukaryotic cells. They have that smooth outer membrane and an inner membrane that's folded into the cristae. Again, the inner membrane creates two compartments: intermembrane space, mitochondrial matrix. Okay, some metabolic steps of cellular respiration are catalyzed in the mitochondrial matrix. And cristae present a large surface area for enzymes that synthesize ATP. Okay, so there's a lot of things that are different about mitochondria compared to other organelles in our cells. If you look at the mitochondria, what internal structure does it possess that make it different from other organelles that we studied? So looking at this picture, what things do you see? Okay, so first there's the two membranes. Okay, second, there's the small circular DNA, so its own DNA, not within like a nucleus of a cell, like in other, in every other cell in our body, called plasmids. So a mitochondria is within a cell that has that has the nucleus with the DNA inside of it. And then also there are smaller ribosomes within there. So those three things are really three differences in mitochondria. Okay, so let's talk about ATP for a second. During respiration, a series of reactions take place in which energy is released from energy-rich molecules like sugars and fats. And you guys have learned about this in STEM. Most of this energy is transferred to ATP molecules. Okay, so here you see ATP, okay, adenine triphosphate. So you see the adenine molecule, and you see a ribose, which is a sugar with a phosphate tail. Okay, ATP, adenosine triphosphate, is the energy, okay, carrying molecule found in all living cells. It's the universal energy carrier. Okay, once made, ATP leaves the mitochondria and spreads rapidly to areas of the cell that need energy. Energy is released by breaking the molecule down to ADP, which is diphosphate instead of triphosphate, and can be recycled back into the mitochondria for conversion back to ATP during aerobic respiration.
Most of a eukaryotic cell's ATP synthesis occurs in mitochondria. A mitochondrion is a sac within a sac. The inner sac is folded, increasing surface area for ATP synthesis. Between the inner and outer membranes is the intermembrane space, a reservoir for hydrogen ions used for synthesizing ATP from ADP. The inner chamber, known as the matrix, is a soup of enzymes that dismantle fuel molecules in the Krebs cycle. The knobs are where ATP is synthesized. Simply put, a mitochondrion is an energy transformer, where energy in fuel molecules such as pyruvic acid is transferred to ATP. Carbon dioxide, the carbon end product of cellular respiration, diffuses out, leaving the cell through the plasma membrane.
Okay, so the endosymbiont theory. So discoveries in understanding the differences between mitochondria and the rest of the organelles that are in cells. So again, mitochondria have their own DNA, which other organelles don't have their own DNA, and this double membrane system, and just the way that the mitochondria functions and the way that it looks led to this theory. So the theory basically states that mitochondria and chloroplasts are ancient bacteria that used to be their own separate organisms, which now live inside larger cells typical of animals and plants. So the DNA and the ribosomes of mitochondria and chloroplasts are still active and responsible for the coding and synthesis of certain viral proteins. But now.
Hi, it's Mr. Anderson, and in this podcast, I'm going to talk about one of my favorite topics in all of biology, and that's endosymbiosis. If you didn't know this, there are two major groups of cells. We have prokaryotic cells, and then we have eukaryotic cells. An example of a prokaryotic cell is like a bacteria. They simply have a cell membrane, cell wall. All of their DNA is organized in a nucleoid region, and they're fairly simple and fairly small. In a eukaryotic cell, we're going to have a nucleus, we're going to have organelles like endoplasmic reticulum, Golgi apparatus, mitochondria. But what we find is when we look in the fossil record, life started about 3.6 billion years ago, and we just see prokaryotic cells for the longest of times. In other words, we don't see eukaryotic cells show up until around 2 billion years ago. And it puzzled scientists how this shift was made, because there are clearly two different evolutionary pathways. The pathway of the small prokaryotic cells and then the larger eukaryotic cells. And they eventually settled on an idea called endosymbiosis. And what does that mean? Well, let's break it down. Endo means within, symbiosis means together, and bio just means living. And so basically we have organisms that are living together within one another. So that's weird. What does that mean? Well, basically, when I read about this the first time, it puzzled me. What we think is way back in the day, we had these aerobic bacterium, ones that were doing cellular respiration, so they were breaking down food in the presence of oxygen. We also had these cyanobacterium that were doing photosynthesis, and they were essentially engulfed by another host cell, and they became the mitochondria and the chloroplasts that we have today. And so that's pretty cool. In other words, these cells became part of a cell and eventually became that cell. That means that the mitochondria that are found in all of your cells are kind of like hijackers that have been inside our cell for billions.
Okay, let's talk about microtubules, structural components of cells. The cytoskeleton is a network of fibers that organizes structures and activities within the cell. It's a network of fibers extending all the way throughout the cytoplasm. It organizes the cell structures and activities, anchoring many organelles into places that they are within the cell. It's really composed of three types of molecular structures. So when you're talking about the cytoskeleton, you're not talking about a single structure. You're talking about three types of structures that work together. You have microtubules, microfilaments, and intermediate filaments. So let's talk about those. Okay, so the roles of the cytoskeleton generally are support, motility, and regulation. The cytoskeleton helps to support the cell and maintain its shape, so it's not just a bag of goo. It interacts with motor proteins to produce motility, so movement. And inside the cell, vesicles can travel along these sort of monorails that are provided by the cytoskeleton. So it provides like a pathway of movement for other organelles. And recent evidence suggests that the cytoskeleton may help regulate biochemical activities. So here's a picture of a vesicle moving along a microtubule. It's fascinating. So microtubules are the thickest of the three components of the cytoskeleton. Microfilaments are also called actin filaments, and these are the thinnest. And then intermediate filaments are fibers with diameters that are in between the two. Okay, so looking at a table for microtubule, they are hollow tubes, which consist of about 13 columns of tubulin molecules. The diameter is about 25 nanometers. There are protein subunits, alpha and beta, which we haven't really talked about those yet. The main functions are the maintenance of cell shape, compressing and resisting girders, cell motility, chromosome movements and cell division, as well as organelle movement. Okay, and you can see what the structure looks like right here. It's this hollow tube. Okay, now looking at microfilaments, and you can see here what the structure looks like, okay, right here. Okay, so the structure is two intertwined strands of actin, each a polymer of actin subunits. The diameter is about 7 nanometers. The protein subunits are actin. The main functions of microfilaments are maintenance of cell shape, changes in cell shape, muscle contraction, cytoplasmic streaming, cell motility, and cell division. Okay, and then the intermediate filaments. Let me get my pen going on here. Red is fine. Okay, so you can see the shape here. Okay, so it's fibrous proteins that are super coiled into these thicker cables. They're 8 to 12 nanometers. One of several different proteins of the keratin family, depending on the cell type. The main functions are maintenancing of cell shape. Again, as you can see, all of the sort of cytoskeleton units are. There's anchorage of nucleus and certain other organelles, and the formation of the nuclear lamina. Okay, so microtubules are hollow rods, 25 nanometers in diameter. We talked about the functions. Okay, let's talk about centrosomes and centrioles, which are an important part of the cytoskeleton. Centrioles are hollow cylinders composed of 9 microtubule triplets, okay, so tiny rings of the proteins. They are found in pairs that lie at right angles to each other. They're used as a starting point for spindle mitochondria during nuclear division, and they are only found in animal cells. In many cells, microtubules grow out from a centrosome near the nucleus. The centrosome is a microtubule organizing center. Okay? In animal cells, the centrosome has a pair of centrioles, each with 9 triplets of microtubules arranged in a ring. Okay, so looking back here. So 9 arranged in a ring around the centrosome. Okay, so here you can see microtubules arranging themselves as a structure in this plasma membrane. And if you zoom in, you'll see here, okay, here is the outer microtubule doublet, okay, one here and one here. Here's the central microtubule, okay, and then so you can see that structure here, okay, and then the plasma membrane is wrapped around this way, okay. So it's what you're seeing here in cross section. Okay, and this brings us to cilia and flagella. So microtubules control the beating of cilia and flagella, which are the locomotor appendages of some cells. And they differ in their beating pattern. So if you look at the flagellum, there's this like propeller-like motion where they kind of whip around this way, like the motion of a screw, where cilia are beat back and forth. Okay. So if you look at, here's a sperm, okay, and it has a flagella that's moving in this almost like spiral circular pattern, and it pushes them, so the direction of swimming this way, it pushes them this way. Okay, so really it's like helping it to move through liquid, and it also helps move liquid over the cell itself. Okay, here are cilia. So you can see the direction of the organism's movement. So you have the body here, and then you have the cilia here. So the direction of the active stroke is moving from this position to this position, okay? And then the direction of the recovery stroke, you can see it's kind of like flapping around a bit, and then that would move it in this direction. And they share a common ultrastructure. There's a core of microtubules that have this plasma membrane. We were just looking at cross sections of that plasma membrane. They have a basal body that anchors the cilium or the flagellum, and they have a motor protein called a dynein, which drives the bending movements of the cilium or flagellum. This is a pretty cool video showing a lot
Many
of
eukaryotic cells have
structures.
highly specialized microtubular structures that extend outside the cell's membrane. These appendages move the cell and move fluids over the cell's surface. The two types of microtubules that perform these essential duties are flagella and cilia. Flagella appear as long whip-like tails on many single-celled organisms, such as this euglena. They also exist on certain kinds of cells in multicellular animals. Some organisms' flagella rotate in a screw-like motion to propel the organism through the water or some other liquid. Flagella often occur in pairs. Some organisms, however, have a single flagellum. Cilia are much shorter and more numerous than flagella. They appear as short, hair-like extensions of the cell. Unlike the rotating motion of flagella, cilia beat backward and forward to propel the cell, as in this paramecium, or to draw fluids over tissues.
Okay, that is all for the cell part 2. Thank you, and cell part 3 is coming up.